Hi everyone, my name is Azza. I am an Odyssey collaborator that spent a lot of time trying to phenotype. And today I'm going to talk to you about this fun phenotype exercise where we try to come up with the phenotype algorithms for the identification and characterization of vaccine-induced thrombotic thrombocytopenia, also known as VATT, in real-world data. This was a multinational network cohort study where I got to work with a group of amazing people from the vaccine safety working group, all who are co-authors on this paper that was published from this work and drug safety. So to give you a background, VATT, or also referred sometimes as TTS, is a safety outcome that has been identified as a rare but serious adverse event associated with some of the coronavirus disease vaccines. So when soon after the vaccine, some of the vaccines were introduced to the public, there were serious reports of patients coming into the hospital with thrombosis and thrombocytopenia occurring at the same time. That was defined as a new phenomena that clinicians did not feel that they knew about from before. So BRIGHT collaboration, which it is the collaboration that comes to identify cases, published an interim case definition for VATT oriented toward the identification and the treatment of cases. But at that point, and even till today, there is yet no consensus or even clear guidance on how to identify VATT cases in observational data or real-world data, including claims and EHRs. So we thought that we need to come up with a phenotype definition for VATT because observational data can be used to generate important evidence such as estimating VATT or TTS background rates and characterizing the profile of patients who had thrombocytopenia with thrombosis in the past. And that's exactly what we wanted to do in the vaccine safety group. So we worked together to and explore the historical trend of the co-occurrence of thrombosis with thrombocytopenia, and that's something I'm going to refer to as TWT to distinguish that from the new phenomena of VATT, because we're not sure if what we're identifying in the past is truly similar to those VATT cases after the vaccine, but that's exactly what we wanted to study. So I'm going to call it as thrombosis with thrombocytopenia, TWT, through an international network cohort study using routinely collected primary care and hospital patient records from 17 data sources across the United States, Australia, Japan, and Europe. And you see in this table the 17 data sources that were part of the study. A lot of those data sources came from, at the time, our new EDEM partners, but now they're all fully integrated within Odyssey. But at that time, it was their first study to work on. So very proud of what they accomplished on this study. So you see that some of this data came from claims, some of it came from hospital EHR, GP records, and we have one registry. I also show you the availability of some elements of the data that are important to identify TWT in observational data. So we expect those cases to happen in an inpatient setting. So I'm showing you the availability of inpatient diagnoses, outpatient diagnoses, inpatient measurements, outpatient measurements, and inpatient and outpatient drugs across the databases. So how did we define VATT or TWT in observational data? So the definition is really based on the idea that patients can basically be eligible to the definition if they have a diagnosis of embolic or thrombotic arterial or venous events and a diagnosis or a measurement of thrombocytopenia within 7 days. At that point, all what we know about these VATT cases that came after the vaccine, that they came to the hospital, very sick people with all different sorts of thromboses, and they have a very low platelet counts. That's thrombocytopenia. We didn't know much other than that, and so we wanted to see did this really occur in the past or not. So we identified through research questions and tried to come up with alternative TWT definition. And this diagram in front of you kind of explain our theoretical thinking behind the research that we did. So the first two main research questions that we had is that what is the estimated background of TWT? And are TWT patients' profile that we observe in observational data compatible with TTS or VATT patients' profiles observed after the vaccine? Those are the two main things we wanted to answer. Now, how did we come out as defining the co-occurrence of thrombosis with thrombocytopenia? So for thrombosis, there are multiple different types of thrombosis that can happen. And so one train of thought was those VATT cases or TTS cases usually have a very rare kind of thrombosis, something like splenic thrombosis, hepatic thrombosis. A lot came with a cerebral vascular sinus thrombosis, a very rare case of stroke in the brain. But then other spontaneous reports coming from VATT cases also showed that they also can come with things like DVT and PE, very common kind of thrombosis. So we wanted to see what we observe in observational data. So the question was, what events make up thrombosis and what is that breakdown? And then we also wanted to know what is the background frequency of platelets count measurements among patients with new thromboses. Because now that this thing is a phenomena, we expected that after the vaccines became available and people are using them, anybody who come to the hospital with a thrombosis who reported that they've been vaccinated, physicians, now that they are worried about this phenomena, will send the patients to get a platelet count or a blood count measurements. And that can cause some surveillance bias when we, in the future, when people are looking at the occurrence of TTS. So we wanted to kind of publish that background frequency of how often do people, when they have thrombosis, get to go a platelet measurement and put it out there. Maybe we could control for it in the future. And then the fifth research question was how to capture thrombosis across databases, or thrombocytopenia, across databases. So some databases have measurements, some databases only have diagnoses, some have a mix of both. So we wanted to see, should we only use measurements? Should we also allow people to be qualified into the thrombocytopenia through a diagnosis? And then if we use a measurement, what is the accurate or the proper cutoff point? Again, in the VATT cases, they were reporting cases of very depleted platelets. So is it 120 per microliter should be threshold point, or is it the 150, which is the kind of common, the used cutoff point for thrombocytopenia diagnoses? So depending on the combination of all these decisions, we came up with 6 different alternative definitions of thrombosis with thrombocytopenia. And then we, using that, we looked at the incidence rate, the background incidence rate, and we looked at patients' profiles. So what you see here is the incidence rate plot of thrombosis with thrombocytopenia using the definition that was feasible across databases. So that is any kind of thrombosis using a broad set of thromboses with a diagnosis or a measurement of thrombocytopenia, depending on the availability of the measurements in the database. So what we found, and this you see it here breakdown by age and gender, and also across different databases, the 17 databases that were in the study. So what we see from the beginning is that the overall incidence rate ranged really widely from 1.62 in CPRD to 150.65 in Medicaid per 100,000 person year. That was important for the clinical community to know, because what people were thinking at the time that the VATT became a phenomena, clinicians believe that the idea of thrombosis happening with thrombocytopenia is just never happened. It's like 0. So we here established that the rate varies and it's not 0. And it could be fairly common depending on what population you're looking at. And then we saw substantial heterogeneity across data sources and by age and group sex within the same data source. And that's clear in the figure right there. And then we saw that there were higher rates were observed among men and older age group. And that was not very consistent with the VATT patient's profile that was published for those patients that had that event after the vaccine. Those patients would tend to be women and young women of productive age. And that's very different from that historical TWT that we observe in the databases. And then also identify cases were likely to have pre-existing conditions such as heart disease, malignancies, and liver diseases, which kind of explain that co-occurrence of thrombosis, thrombocytopenia, while those cases coming after the vaccine were, again, young women, healthy women, have no health conditions, other health conditions, and they were very much younger. So we concluded that thrombocytopenia does co-occur frequently with thrombosis in the real-world data, unlike what was clinician thinking at the time of the phenomena. And the current definition is likely to capture cases that are not a true representation of the new emerging clinical phenomena of VATT, but merely a coincidental co-occurrence of the two common clinical events. So we thought that what we're observing is just a kind of a chance of the co-occurring of the two events, given that thrombosis itself is fairly common and thrombocytopenia are common, so they can co-occur within 7 days or so, and so forth. And that we cannot rely on generating background rates of bits from observational data, again, because of that noise. And then if real-world data or evidence is to be used to explore, we need other further evaluation of cases will be required. And then finally, my conclusion was that the vaccine safety work group are really the best team to work with. Thank you. That is my last slide.